Welcome everyone. Today we have our own Dr. Schonbrun here. He received a PhD here at CU, and about 3 years ago he left to Professor Ken Crozier's group at Harvard, and now he's a research scientist there, and he'll tell us some cool things about optofluidics today.
Okay, thanks. Okay, nice to be back in Boulder. I'm going to talk about measurement platform and optical measurement platform that we've been working on, a couple different generations of these devices that we've been making. Integrations of microfluidic and micro-optical components. One of the main applications that we're going to target is high throughput measuring of biological samples. There's a couple of devices that I'll later talk about more. So, so one of these major applications that we're going to target is high throughput analysis for biotech. There's a variety of applications in biotechnology when you want to measure a very large number of samples. So one of these is for sequencing DNA. For example, if you want to sequence the human genome, there's many billion base pairs in the human genome. You need to measure them several different times in order to assemble them back together again, so you have to measure tens of billions of measurements, often labeled by different fluorophores. And so if you can figure out a high throughput method to do this, very parallel and very fast, you can make this process cheaper, more uniform. Be able to do this to a broad number of people instead of just a few. Other applications when you want to do high throughput analysis is if you're looking for something that's very rare. If you're looking for a cell that only occurs 1 in a million times, you have to measure many millions of cells in order to get reasonable statistics. And there's also applications in drug discovery, or they often do combinatorial chemistry, where they want to mix different chemicals in a very large number of combinations, looking for whichever one produces the reaction that they want, and then they send it through feedback. And you want a method that gives that feedback that tells you what chemical reaction is happening. So here's two different optical measurement systems that are used in this field. Both are very successful. They perform slightly different functions, but they're both able to measure a large number of different samples fairly quickly. One of them is a microarray scanner. This is just a microscope slide, like 3 inches by 1 inch. You can print different samples onto them, different sequences of DNA, for example, with which to measure your sample against. And then you read out the fluorescence that occurs on this microarray. The microarray scanner is just kind of a scanning microscope that reads out the dots, mixed up mechanically, and, optically and mechanically. There can be anywhere from tens of thousands of those dots, so the density can be as large as a couple million. In the case of a couple million, you have like a dot every like 20 microns or so. And it will take maybe a couple to 30 minutes to read this entire slide, which has all this data on it. So that's a popular technology. Another one that is maybe more similar to the one that I'm going to present today is flow cytometry, which uses a mix of fluids and optics. You send your sample into what is often a capillary tube, and then surround it with this other fluid, and then through this process of geometric confinement and hydrodynamic focusing, you can measure particles in this fluid, often cells, therefore it's called cytometry, but it can be a broad variety of different particles in suspension of this fluid. You engineer it so they go by one. You can measure as many as tens of thousands of samples per second. State of the art is maybe 50,000. So it's a very high throughput technique. There's different optical signals that you can measure. You can measure the forward scatter, side scatter, but the most common data is often just fluorescence. Measures different Sometimes you can measure as many as like several fluorescent colors using a combination of dichroic filters. Here's an example of a flow cytometer. It takes up an entire bench, so it's capable of this high throughput and very successful measurement platform. So our technique is very similar to flow cytometry, but we're going to read it out in a method that looks a little bit like those pictures of the microarray. So the platform that we're working on is microfluidics. It's a planar fluid manipulation platform. You define these circuits with lithography, so they're very flexible. There's very easy to parallelize, very easy to make small. So you make these microfluidic channels. They can have dimensions on the order of several microns, but these have widths of 20 microns. So this device has a pitch between the channels of around 30 microns. The first device that I'm going to show is a droplet-based microfluidic system, and then I'll show some more that are not droplets. But this droplet-based system is a two-phase microfluidic system. The inner phase is water, and the outer phase is oil. And what is commonly done is you put your biological sample into the water phase, and the water droplet acts like a discrete container. You can process and transport an enormous amount of these containers through your system. I'm going to focus mainly on measuring what's in these containers, but there's a variety of different ways to manipulate them. You can split these droplets, you can recombine them, you can mix what's inside. One appealing feature of these drops is that you can produce them very quickly. So they're formed in a flow-focused drop maker. I have a slide at the end that I can show if you want to see how that works. But these water droplets are basically spit out of a nozzle at rates of anywhere from hundreds a second to several thousands per second. And then we can parallelize this. So the device that we fabricated was a single flow-focused drop maker. It was then split many times, 64 times, to create an array of these drops with which we want to measure and detect what the fluorescence inside. But if you want to, so traditionally the fluorescence is read out of these drops just by focusing a single laser beam onto a single channel and just measuring the fluorescence that is both excited and collected through an objective lens. But if you parallelize the system, you end up with a large area device, and it's difficult to efficiently capture the fluorescence over this large field of view. And this is a movie, so if you want to see this slowed down by a factor of 50, and this is not running at maximum rate. But so these droplets are formed over here. They move through the system, and we want to measure the optical properties in these very small containers, and these droplets can be produced, thousands of drops per second in each channel, multiplied by how parallel this system is. And we're going to use an integrated lens array to read out the signal in this array of microfluidic containers. And this lens array can help us solve this problem of needing to detect efficiently over a large field of view in an integrated system that will hopefully be much simpler than those large flow cytometers and potentially much faster as well. So I've been working on high numerical aperture diffractive lenses. Before this project, we were working on using high NA zone plates or diffractive lenses for optical trapping. For optical trapping, you want very large NA, so diffractive optics was interesting. They're also interesting, we believe, for this project. I'll talk a little bit about some of the benefits when we're using diffractive optics versus other kinds of micro-optic systems. But a simple one is just that you can make them out of many different materials. We were doing some surface plasmons in the beginning, so we started with metals, but very quickly we went to semiconductors. This one's made out of silicon. Those are nice because the index is large and you can get a high phase shift for a very thin structure. But for microfluidic systems where the fluid handling system is often made out of either plastic or glass. Making them out of plastics that we can mold is very appealing. So the lenses that I'm talking about in this talk are going to be made out of PDMS that we make with a soft lithographic process. And then future designs are going to take advantage of the fact that you can pretty easily integrate these systems with various holographic functionality. These are just two designs that we made for the trapping project, which this lens creates 4 focal spots that is able to trap 4 particles simultaneously. And this lens, so the trapping laser was 980 nanometers, and we designed a lens that would focus 980 nanometers to the same spot as the fluorescence emission, which happened at 575 nanometers. And normally these diffractive lenses have a lot of chromatic aberration, but when we superimpose these two designs on top of each other, we could focus and collect from the same region with these 2 different colors. But anyways, it just shows that there's a lot of flexibility that goes along with working with diffractive optics, and that they're good. It's a good it's a good system to integrate into MEMS and microfluidic systems. So I just have a couple of slides about what we were looking for in the design. This just very simply shows that previously we were making very small lenses, but in order to efficiently focus and collect light, it makes sense to have the lens not be so small. And if you think about, we're going to use the lens to both focus light into the channel, and we're going to call that an excitation enhancement. And then we're going to use the lens to collect the fluorescence emission, which we're going to call an emission enhancement. And both of these roughly scale as the area of the lens itself. So if you want to transform the optical intensity incident on a lens into an excitation spot, and you look at how bright that excitation spot is, it's roughly the ratio in area times the efficiency of the lens. Scales as the area. And the emission, if you consider that you have an isotropic radiator, it also scales paraxially with the area of the lens. So we want to make our lenses large. If you multiply these 2 enhancements together, it just scales with the raised to the 4th. So we want to make our lenses larger than the channel itself. Let me just talk a little bit about the geometry. So this is the geometry of the system. I'm going to talk a little bit about the fabrication, but both the lens array and the microfluidics are made out of this polymer, PDMS, and the two systems sandwich a cover slip. So we design the lenses to have a focal length of a little bit longer than the thickness of the cover slip. The lens has a width that's much larger than the channel width, so therefore it can collect a lot of energy with which to focus into the channel, and then when the fluorescence emission in the channel radiates out, it can be efficiently collected about as high in a zone plate. So we use a #1 thickness cover slip. Has a thickness that we measured of 180 microns, and the width of the zone plate is around 200 microns. The focusing occurs in glass, so it's kind of like a solid immersion lens, which helps with the numerical aperture as well. The numerical aperture is the index of the immersion medium times the sine of this angle. The design that we often use in this talk gives us a numerical aperture of 0.8, which is fairly large. And then just this is a little bit more about the design of the lens. You can consider these diffractive lenses as just a hologram, where the reference wave is a plane wave and the recorded wave is just a spherical wave. And the origin of the spherical wave can be located anywhere transversely, x naught and y naught. But the interesting thing is it can be as far away from the recording material or where you place the lens. This distance is arbitrary, so this is the origin of the spherical wave, and here would be where the lens is. And because we can place this very close, we can make the focal length as short as we want. So we can make the numerical aperture very large. You can make the numerical aperture as large, basically, as the index of the medium that the spherical wave originates in. And another interesting aspect of these large numerical aperture diffractive lenses is that they don't have higher order focal spots. Normally you think of a diffractive lens that has two layers creating very many focusing orders. But when the numerical aperture is large, the only diffracted waves are a wave that focuses to plus f, and then one that diverges at minus f. The higher orders are evanescent, which makes the far field very clean. And because the focal length is very large, the focal length is very small, and the numerical aperture is large, the lens has lots of power, so the diverging wave diverges very quickly and its intensity is low. And then additionally, if you make the zone plate have a pi phase shift, there's no zero order wave, and you just have these two very quickly converging wave and very quickly diverging wave. So the diffractive lens doesn't have perfect diffraction efficiency. If it has a pi phase shift, it's 40%, but there's no other orders to worry about, which would happen for a lower numerical aperture diffractive lens. Please ask any questions. Okay, so this is a slide about the fabrication. So what's cool is we can make these zone plates with the same procedure that we make the microfluidics. For the most part, in the fact that we can mold them. The one difference is that so here's a picture of the zone plate. This is 200 microns. The rings get closer together as you go further out, and at the radius, the pitch is around 700 nanometers. So the width of each ring is like 350 nanometers. That's a little bit smaller than we can make with optical lithography. So we spin on a film of SU8, which is both a photoresist and an E-beam resist, but we use it as an E-beam resist in this case. We pattern this with electron beam lithography, develop it, and then this is the master. And from this master, we can mold replicas very easily. If you make a very large lens array, it can take as long as several hours to write that pattern with the E-beam, but once you've made your master, you can mold it many times. And you can even use the molded lens array several times. We'll talk a little bit about how we align and adhere that to the microfluidic system. So we spin coat a 500 nanometer layer. We exposed it and developed it. We got features that were approximately 400 nanometers deep. Ideally, we would be 600 nanometers deep. 600 nanometers would be pi phase. We're like at 2/3 of pi phase. Approximate diffraction efficiency is 30% instead of 40%. But the fidelity of these lenses is pretty good. Side walls are pretty straight. And, the depth is pretty uniform across the radius. So this is just a short schematic of the experiment, how this is going to work. So this is our two-layer kind of fluid optic system. And how we're going to operate the system is we're going to illuminate the entire lens array with a large excitation laser. So, and I guess the largest array we did was an 8x8 array. So that array is 2 millimeters in total cross section. That's about the size of a laser coming out of the beam coming out of our fluorescence excitation laser. We introduced that with a dichroic mirror, and then just with unity magnification relay optics, we image the lens array onto a camera. So we're actually not imaging what's inside the channels, we're just imaging the aperture of the lens arrays onto this fast CMOS camera. And when there's something interesting, when there's fluorescent molecules inside the detection region of these zone plates, the aperture of these zone plates lights up, and we see that by looking at their image. I'll talk a little bit more about this. So we can characterize the excitation and the collection enhancement of these lenses, because even without these lenses, these relay optics can still image what's inside of these channels, but with very low efficiency. The numerical aperture of these is around 0.1. So we can still image what's inside of these. Have a very large field of view, right, because the magnification is so small. We can still image all the channels at once with just the relay optics, but we can compare the signal with just the relay optics to what the relay optics do in conjunction with the zone plates. So we can characterize the excitation enhancement. We just illuminate a lens, have the lens focus excitation light into the channel. It creates fluorescence emission, and we image that spot with another microscope. We get a spot like this, around 15-20 microns in cross section. So we've designed the focal length of the lens to be a little longer than the cover slip thickness. So this is not diffraction limited, it's defocused, but actually has a width that we want. We want the width of the excitation spot to be roughly the width of a channel, so that our channels are 15 microns, so it's not exactly flat. I have some slides at the end of trying harder to make that excitation flat and uniform across the channel. But so this is the excitation spot. It has a peak intensity of around a little less than 20 times as compared to without the lens. That's about the same as the ratio of areas times the diffraction efficiency. So that's what we expect. We can also characterize the emission. So we characterize the emission by doing the opposite. We focus an excitation laser with a microscope objective, scan that excitation spot, which creates this emission point source. We scan that spot across the optical axis of the zone plate, and we see what we thought originally was an interesting effect. So there's two things that we can measure here. One is how efficiently the zone plate collects the light, and secondly, where the zone plate collects the light. And what we find out is the zone plate only efficiently collects the light when it's overlapped with the optical axis. So when the point source is overlapped with the optical axis of the zone plate, and what we get is we get a curve like this experimental curve also, and we can see that when it's right on the optical axis, the whole zone plate lights up. And so what's happening is, there's two different ways of looking at it. It's similar to a confocal filter. It's also similar to a vignetting effect, where the zone plate is only efficiently collecting light in a small detection region, and when it's outside that detection region, it's being filtered out by the aperture of the relay optics. But we can get enhancement of around 12 times, as well as measure the detection region, which is a function of the aperture of the relay optics. But if we get an enhancement of 12 and enhancement of 20, total enhancement multiplied together around 200 compared to just the relay optics alone. I think I'll skip this slide, but one more thing I want to mention is, so we have this confocal filter effect, but the nice thing is this happens in parallel for all the lenses in the array. So all the lenses in the array have their own confocal filter, and they all collect light only from a well-specified detection region, and this reduces crosstalk in our device. The lens only looks at the fluorescence in the channel that it's spent. Okay, so now I'll put this all together and show a couple examples of devices that we've made. This is one of these treed out microfluidic systems. The channel spacing is around 200 microns, which in this case is the same as the diameter of the zone plate. The diameter of it's a little less, but around that same order. We illuminate all of these zone plates with an excitation laser, image them onto a fast camera, and then if we put a lot of fluorophore on the drops and run the camera very slowly, we can actually see the drops even when they're not in the detection region of the zone plate. So these are drops that are just being imaged by the relay optics alone, but when they go through the detection region of the zone plate, they'll flash, they'll light up because of that 200 times enhancement, and this is what they look like. So, this intensity is 200 times higher than this, and then when we run this camera fast, and we reduce the amount of fluorophore that's in each drop, you can't see these guys anymore, and you can only see the drop when it is in the detection region of the lens. So this is our improved geometry. In this case, we wanted to pack the microfluidic channels much tighter together. We didn't want to space them by 200 microns. We wanted to space them by only 30 microns, but we wanted to keep the lens diameter large because of the importance of the large radius and the large area of the zone plate. So this is this takes a single channel, splits it up into 64 channels, and this is an 8x8 lens array. And that we found by tilting the lens array by this angle alpha, we can actually have the zone plate pitch be much larger than the microfluidic channel pitch, which allows us to keep large lenses even though there's very tight spacing in the microfluidics. Just a simple trig will tell you what angle that needs to be. And then we can run all 64 of these in parallel. And then we can go pretty fast. So each channel can, we're producing drops at a rate of 3,000 per second in each channel. We have 64 channels. This is not that, this is running a little slower than our max, but this is what the movies look like that you can collect. And then, so you collect this huge image stack that you can then later analyze. This movie is slowed down by a factor of 50. I think this movie is showing drops being produced at a rate of 20,000 per second, so we could go another 10 times faster. But the signal-noise ratio isn't as good. So the signal-noise ratio is good here. That's why I show it for a movie. So we have this image stack. We can analyze what's going on in each channel. And in our case, every drop has fluorophore in it. So we get this periodic train of signal. In this case, the drops are coming at a rate of 3 kilohertz, and we're sampling at 16,000 frames per second. So every 5 frames, fluorescence peaks, and we can pick any of the 64 channels to analyze. So we were able to measure 200,000 drops per second in this device, but we're both in time and space sampling, not at Nyquist, so we think we have a reasonable room for improvement. These very fast CMOS cameras have enormous, you can say pixel throughput, how many pixels that they can collect per second. So we did a subarray where this is, I think, forget actually, they're 64 or 128 by 64 or 128 pixels at 16,000 frames per second. I think we were collecting 100 million pixels per second, but these cameras can run up to several billion. So if you stuck a single pixel on each zone plate and then had a dark one in between for like Nyquist, and then these drops are nice because they're very periodic, so you can also sample in time at Nyquist. You could potentially build a system that could measure getting close to 1 billion entities per second if you were trying to do Nyquist in all those 3 dimensions. Okay, so I talked a little bit about, so we made this round spot previously. We then did a little work to try to create a more uniform excitation profile across the channel. So we added some stigmatism to our zone plates, which was, so instead of just having, so this is just your normal zone plate equation, we added a cylindrical lens factor. Now we have a stigmatic zone plate that has 2 focal lengths, has a front one and a back one. You can calculate where they are. The back one is just where the circular one is, and the front one has to do with how much focusing power you give to the cylindrical lens. And this gives a line excitation. Line excitation is both narrower along the direction of flow and has a more flat intensity distribution transverse to the direction of flow. So I made this. They work as expected. This is the front focal plane. This is the back focal plane. This is a device that we made. This is a better picture of this flow focusing drop former. And then we aligned 3 zone plates. So instead of making a parallel device, we just wanted to detect what's going through the channel at 3 different times. And what we really wanted to do was try to make, so instead of measuring these drops, we sent beads in. And the beads are much smaller than the channel cross-section, so they can kind of distribute themselves quasi-uniformly. And we were finding that with our previous zone plates that the signal was very different if the bead was in the center versus in the side. So we tried to see if we could fix that with optics by creating this more uniform intensity distribution. So this is filling the channel with fluorophore. We get 3 lines. And then if we compare, say, if we send 1 micron beads in, we want to make sure that each zone plate collects the same signal because we know that the beads are the same. So that if we're sending in a more interesting sample like cells of different sizes or beads of different sizes, and we measure a different signal, we can actually trace that back to the bead as opposed to just not knowing if it's in the center of the channel versus the side of the channel. And so we did this. This is 2 micron beads, 2 micron diameter beads. You see that the uniformity of the signal is pretty good. There's around a 20% standard deviation in the size of the signal from each zone plate. And then a bead is always measured. Very few beads are missed. So in the previous device, we were finding that maybe 1 zone plate would measure the bead, and then the bead would like go around the excitation region of the next zone plate. We don't find that here. We find that I wrote like a peak counting algorithm, and the number of peaks that each zone plate counted was the same, which tells us that the signal is uniform. So another interesting experiment we can do with this kind of different device, that instead of running it in parallel by measuring many channels, we're making many measurements along the same channel, is we can do velocity measurements and we can do velocity dispersion measurements. We can compare the signals from different zone plates, and if you, okay, so similar to particle image velocimetry usually takes a single microscope, and inside the field of view cuts it up into regions and correlates the regions together, and then based on that time delay and the distance between them, you know the velocity. So you're usually limited by the size of the field of view of that microscope objective. So what we're doing is each field of view of the zone plate, we don't have a whole image, we just have a spot, but we have what is potentially a very large field of view because we can correlate the signal from different zone plates, and the distance between these different regions can be very large. So we can measure what happens to the beads as they travel through these channels. And so when we do correlations, we can correlate the signal between like 1 and 2, and 1 and 12, etc. And we get data that looks like this. So this is just the time delay. This is the strength of the correlation. The location of this peak tells us the velocity, and then the size of the peak actually tells us something more interesting about the distribution of velocities. If all the beads are traveling at the same speed, these will still be delta functions, but in fact the beads are not all traveling at the same speed. They have some distribution, and we can measure that distribution more carefully if we choose. So the fluid in a channel has a parabolic flow distribution. The beads in the channel have a more complex distribution. They can do a variety of different things. In the laminar flow regime, the beads kind of have a similar distribution as the liquid themselves. So the liquid itself has the highest velocity right in the center, and then it actually goes to 0 at the channel wall. That's what people call the no-slip condition. Beads can't get infinitely close to the channel wall, so their minimum velocity is not 0. But we can see how this, so here are 2 correlation peaks. The peak, most of the beads arrive at the very beginning, and then as this has a very slow tail because at close to the wall, the velocities are very slow, and that time delay takes very long. So we can resolve this decay, and this tells us something about both the velocity of the beads as a function of position and the distribution of the beads in the channel. And at larger Reynolds numbers, we find that most of the beads are not in the center of the channel. They find some happy medium next to the wall, and that these peaks don't look like it's very asymmetric shape anymore. Most of the beads take some intermediate time to arrive at the next detection region. So anyway, in summary, we've been working on this new platform for making fluorescent measurements. We mainly been focusing on trying to make very high-speed, highly parallel measurements, which we think diffractive optics lens arrays have something to contribute to. They integrate well with these parallel microfluidic systems because both the materials are similar and their lithography allows us to shape and control the location of both systems very accurately. We've shown a system that's pretty similar to flow cytometry. It doesn't do all of what a flow cytometer does quite yet. It does measure several spectral channels and things, but we think we have some ideas how to measure different spectral channels with diffractive optics. But it is able to go very fast. This is a factor of 4 or 5 faster than state-of-the-art flow cytometry. We've shown that we can add some holographic functionality. Initially, we just added some stigmatism, but there's a variety of different other things we could do by adding some holographic functionality into the lens design. And then we've begun to study some fluid dynamics, both with the dispersion of velocities of beads and channels, and we might in the future add some more interesting geometries into the system. Yeah, so that's it.
Questions, please?
Can you talk about replicating these zone plates that have 300 nanometer wide but 600 nanometer deep? Those are deep trenches.
Yeah.
Is it hard to replicate them and pull it out such that that feature is reproduced?
Yeah, so 2 to 1 starts getting hard. So we weren't actually able to make, so 600 nanometers gives us pi. We can do 2/3 of pi, which is 400 nanometers.
But I mean, does every ring come out?
Every
or is there
ring,
some
every ring will
statistics?
come out.
Every ring comes out. So how hard is, how do
I
you
would
do
say
that? Tell us how to do it.
Several out of 1,000 don't come out. Yeah, so yeah, PDMS is this amazing material that wets the surface and is able to kind of dig itself into any trench. And then, yeah, so it's actually very simple. It's just you mix this PDMS and 10% of the curing agent. Sometimes we put, after we made our master, we can you silanize it, which allows the PDMS not to stick very well. You just put it in a vacuum chamber for a couple hours with a little bit of silane. Then you pour the PDMS on it, bake it overnight, peel it up. I mean, it will work the second time you do it, if not the first.
And this is just done in SU8. It's not like a metal master. Does the SU8 wear out doing this?
Like how many replicas could you make? I've never made more than 4 or 5 from the same master. Maybe you need to re-silanize it, or I don't really know if you want to make 1,000 how it would work, because I want to change the design after a couple times. But certainly several, I don't see any degradation.
So when you were actually detecting a signal, you were imaging the full aperture of the zone plate?
Yeah, just imaging the zone plates onto the camera.
Yeah, I was just thinking for a detection problem, it seems like that would have lower SNR than just integrating all that with another lens. Right, so like if you like focus that image, you would have a higher SNR, and I would think you might be able to get a higher throughput. Is that a problem to do that?
Put a single pixel onto the image.
Well,
I kind of am integrating it onto.
Okay.
I mean, these. I mean, so this image is taken by I take my image stack and then I just add like 1,000 frames together, so at least, yeah, anyway, so there's like several pixels inside each image of the zone plate. And then I integrate all these pixels to get these signals.
Yeah, I was just thinking like if you had like some additive noise, like just Gaussian noise or something like that, that just doesn't seem like the optimal situation versus just integrating all that onto one pixel.
I don't really understand. Integrating this entire image onto one pixel?
Well, like if you took a, I mean, like, so all I was asking is like, so you have all this light that you're imaging the zone plate aperture, and what if you could just have another array of zone plates that focus that aperture back down onto a single pixel or few pixels?
I can't put it on a single pixel because I need to resolve. I mean, I could potentially put this on 16 by 16 pixels, but each of these zone plates is observing a single channel, and I don't want to mix what's happening in one channel from its neighbor.
Oh, okay, so you're just worried about.
I mean, it's not really creating an image, but in a sense, I am spatially resolving the different channels. So maybe I don't understand.
Yeah, I think maybe I'm not saying what I want to say correctly, but I know what you did.
There might be a
I
better way.
understand
I don't
what
know.
you did.
So here you're using an 8x8 grid, but you discussed using a single zone plate to create multiple spots.
Yeah.
What are the trade-offs as you go from multiple zone plates, each
Yeah.
an individual single zone?
I was kind of hoping to do something like that, like a single zone plate, but just naively, the first design is if you have a single zone plate creating many spots, and imagine a point source occurs in one of those spots. I didn't know how to figure out if it was occurring in one versus another. And you could create many excitation spots very easily with a single zone plate, but figuring out where the light was coming from in collection, I didn't know how to solve that.
Could I ask a follow-on?
Yeah.
When you did create multiple spots, did you see any reduction in diffraction efficiency?
It was pretty good. I mean, the diffraction efficiency in each spot was pretty close to 1 over the number of spots. Yeah, when I superimposed the 2 wavelengths, that one didn't quite work as well as theory said. But,
how do you load your molecular samples into the droplets? How do you make sure there's one species?
yeah, those are good, difficult questions that people are starting to address. Yeah, so I did this work in collaboration with the microfluidic group, and they've done a lot of work on loading beads and cells into these drops. And there's a variety of things you can do. If you just do it the dumb way, you get some Poisson distribution of how many entities you end up in each drop. But there's certain things that you can do that do beat that distribution. If you, yeah, we can talk about that more later. But like order of the loading. And the second question was, do you get something interesting into the drops?
Well, how do you make sure that you only have one, either one entity or one kind of entity?
Yeah, I mean, you get some statistical spread. If you're making 1,000 drops per second, you won't have 1,000 drops that each have one. But so flow cytometry, which is an established technique, also addresses the same problem. And what they do is they have some nozzle that creates, like, sometimes there's a variety of ways to do it. Sometimes it creates water droplets in air, and I think it creates, like, these are small. They're probably twice the size of the cell, so 20 microns. And it probably creates them at like hundreds of thousands of drops per second, and then they load it in such a way that 1 out of every 10 or 100 drops have something in it. So that will follow the Poisson distribution, and very rarely will they ever get something that has 2 drops in it, although that will happen sometimes. So they They do it by having most of the drops be empty.
When you're designing these microlens arrays, it seems like you generally design one array to match with one fluidic channel design,
Yeah.
right? So I mean, are these fluidic channels, like, can you reuse them many times, and do you have to, can you just make your fluidic array and your microlens array and just align them once and stick them together and you're done, or do you have to change that each time you're, say, testing different species of things or making new samplers?
I think if you just want to load something different in the microfluidics, yeah, nothing has to change. You have some system that works. Yeah, and a new microlens array will have to be designed around the fluidics. So if you want to change your geometry of fluid channels, you're probably going to need not like a one-size-fits-all for the. But you can.
I mean, I guess it just seems like the alignment of your microlens array and your fluidic channels is, you're obviously sensitive to that. So
Yeah. The alignment actually wasn't that hard. What's harder is, especially with these results at the end, where I wanted the signal from a bead to be as uniform as possible, so I could in the future actually compare signals, because we have some ideas to make zone plates sensitive to different colors or maybe different shapes. In that case, you want to maybe compare the signal from one zone plate, which is looking for one thing, the signal from another. So you want this variance to be 0. The depth was hard. Like I was sitting there measuring thicknesses of cover slips, and I'm not even sure that they were that flat. There might have been some wedge angle, and if I'm making my focal length, so in the case with the circular spot, I just defocused the light a little bit, and that was fine. But with the case with the cylindrical spot, I really wanted that front focal plane to be exactly at the right depth, so I was sensitive to depth. So that alignment was actually hard.
The flow cytometer can not only detect those, but also sort.
Some can, yeah.
Can you do that here too?
I haven't done that, but this group that we collaborate with has done a lot of electrophoretic sorting, and you can actually embed soldered electrodes into PDMS. And they've sorted with a single channel at rates of several thousand per second. And they do the same thing. It's fluorescent activated cell sorting. They call it FACS sometimes. And you look for a fluorescent signal, it flashes, and then you turn on your electrodes. That would be hard to parallelize with 64 channels, I think, but this system can support that potentially.
When you said that the zone plates don't have multiple orders, I can see why that's true on the outer rings,
Yeah.
but the inner rings seem like they would.
Yeah.
So
It's
they,
not rigorously accurate, maybe. That's true. When the NA is really high, the period quickly approaches the wavelength. So for the zone plates that I was making for optical trapping, which had a numerical aperture of maybe 1.28 in water, most of the periods had spacing less than the wavelength. Less than 2 times the wavelength. But for these ones that have a numerical aperture around, there's more of the rings in the center. That's true. It's not exactly.